Christina is a research director at INSERM, and she leads a team at the Institut de la Vision in Paris. So originally from Germany, she studied chemistry in Freiburg, in Seattle, and Berlin. She obtained her PhD in chemistry at the Free University of Berlin in 2004, which focused on the identification of a new gene involved in congenital stationary night blindness. And after her thesis, she continued to work in the same retinal disease and identified 3 other genes involved. In 2007, she was recruited as a team leader at the Institut de la Vision, and in 2010 as a permanent scientist at INSERM and research director in 2015. So today, Christina is going to talk to us about visual restoration in mice with CSNB. Christina, thank you very much for accepting our invitation, and we look forward to your talk. We can see your slides.
I think you're still muted.
But we can't hear you.
Sorry?
Yes. All good.
So still muted?
No, we can hear you.
Okay. Now it's working?
Yes.
Yeah, sorry for this. So not everybody knows what it is, congenital stationary night blindness. So I will give you just a brief introduction about this. So it's a clinical and genetic heterogeneous retinal disorder. The prevalence is unknown, but it's largely overlooked. So, and this is mainly due to that you need specific clinical examinations to correctly diagnose this disorder, like electroretinography. So, however, we want to have an idea about the prevalence, and therefore we checked our cohort, at least, which was molecularly diagnosed. And we found that it is a rare disease with only 1 to 400,000 affected. But however, I think we really need to diagnose correctly this disease, and then the numbers will increase. So the degree of night blindness, also the name implicates this, that all patients have night blindness. It is variable. Some people even do not complain about night blindness. You need to really ask the good questions to know if these people have night blindness. A lot of them are surprisingly more photophobic, so they avoid light, and others are very highly myopic. Also, hyperopia can occur, and patients have often nystagmus. This is one of the first signs where parents can diagnose this disease and will then visit a clinician, because the patients are night blind from the beginning, from birth, like the name implicates. So often patients and parents do not recognize that patients, that their children have this night blindness. Then also strabismus may occur, and also surprisingly, the visual acuity can be really reduced. It is reported to be stationary, but there are also cases of slowly progressive disorders associated with night blindness, and then this leads to slow progressive rod or cone degenerations. So genetics and also in vitro and in vivo studies by us and others have helped actually not only to correctly diagnose molecularly patients with this disease, but also have an idea about where the proteins are localized and about the signaling from the photoreceptors to the bipolar cells. So you can see here molecules in green, and they are important for the calcium influx at the synapse of the photoreceptors, which then subsequently, so this is the alpha subunit of the calcium channel associated with other proteins. Jan Kremer, he was talking about a mouse lacking, for example, this calcium channel. And these molecules are important then for the calcium influx and then for the glutamate release, and subsequently the mGluR6 or metabotropic glutamate receptor, also called GRM6, takes up this glutamate, and then this leads to a closure of this TRPM1 channel at the end of the bipolar cells. And when you have light, subsequently the glutamate gets decreased, and this will lead subsequently to the opening of this TRPM1 channel, and this leads consequently to this B wave Mirella was talking about, Jan was talking about, which is then absent actually in patients with the complete form of CSNB, because these patients, they have mutations in all these molecules which are mainly localized at the synapse of the dendritic tips of the bipolar cells. So they can't, the glutamate cannot be taken up anymore. These are all loss of function mutations in patients with complete form CSNB. And then there are also another form called incomplete CSNB, like mutations in CACNA1F lead to incomplete CSNB. And they are mainly located at the synapse of the photoreceptors. They locate, for example, at the rod and the cones. And since rod and cone synapse was on and off bipolar cells, here we have an on and off dysfunction, where patients with complete CSNB, they have only an on dysfunction. Yeah? So this is very important. And if you have good clinical data, then this helps to perform the molecular diagnosis rapidly. You don't need to do whole genome sequencing to identify and subsequently a CACNA1F mutation. So today my talk will concentrate on complete CSNB. And I want to tell you that, of course, these genetic approaches are helpful for the patients, but also we and others developed mouse molecules, because when we identified by whole exome sequencing mutation in GPR179, nothing was known about this molecule. So where to place it? We found by RNA in situ hybridization studies that, yeah, it's in the internuclear layer, but where exactly? And does it interact with mGluR6, which was known before, or with TRPM1 or NYX? So therefore, a mouse model is very important, because you can then subsequently localize this protein in the cascade. So when we don't have GPR179 in this knockout, then subsequently this leads also to a mislocalization of these proteins, these regulatory protein important to shut off or for this cascade. Similarly, when we have LRI3, we found out that when we don't have LRI3, also TRPM1 is gone completely in the on-cone bipolar cells as well as in the rod bipolar cells. So we knew LRI3 needs to be in close vicinity to TRPM1. And similarly, however, also, which is interesting, there might be a double role. Similarly, also other proteins were gone, but only in cone on bipolar cells, namely GPR179 and mGluR6. So, but mouse models, of course, help as well to develop therapies, and therefore we were interested to make an AAV approach to restore the protein localization of mGluR6 and LRI3, and of course, hopefully as well, restore the function of the B wave in these mouse models. This was mainly the project of my former PhD student, Juliette Varin, and she performed a lot of ERGs and AAV injections, intravitreal injection, to restore this phenotype of mice lacking GRM6 and LRI3. You see here, mGluR6 is mainly located in the dendritic tips of rod, which are the tiny dots here, and cone on bipolar cells. And also TRPM1 co-localizes with GRM6, also called mGluR6. And in a mouse model lacking GRM6, luckily we don't have any GRM6 anymore, no dots, but also other proteins of the cascade like TRPM1 are severely reduced. And in addition, it is very convenient because CSNB, also the mouse models, they don't have a B wave. But as patients, the retinal function in patients with complete CSNB, and also in mice, the retina is in a quite good shape. So we thought, why, this is good, because we could even treat adults, adult mice or later patients, because there is no degeneration of the retina. So Denise nicely prepared us vectors. This is her grade AAV7M8 vector, and which we used then for intravitreal injections at P30, so in adult mice. And we wanted, after a couple of weeks, check if the protein GRM6 and other proteins of the cascade relocalized, and also if the B wave can be measured. And in addition, we have done behavioral studies as well as multi-electron assays to investigate if the on pathway is restored. So Denise made these vectors. She had made this nice figure to show that we wanted to target bipolar cells by using a GRM6 specific promoter, including, of course, the whole gene, the coding sequence of the gene. And at the same time, we tried to target the whole retina with a ubiquitous promoter and GRM6. And this led to the following results. Here you can again see the absence of the protein staining in the mouse lacking GRM6. But when mice were treated with the AAV promoter GRM6 vector, so targeting bipolar cells, the protein could be nicely relocated in the dendritic tips of bipolar cells. At this point, we don't know where, in the rods only, or also in the cones. We don't have these bars, you can see this already. And then similar results were actually obtained with the other promoter, with targeting the whole retina. Then a global view of the retina shows you nicely that, however, it worked, but however, it's more locally, the localization after the treatment. In the CAG treated mice, it looks more broad, but also other layers were showing afterwards embryonic staining. Our results then showed that when we compared or checked the transduction efficiency for the whole retina, that only 2.5% targeting the bipolar cells were transduced, and only 11% of the retina was transduced when the ubiquitous promoter was used to target the whole retina. So, however, we could also identify localization of TRPM1 after the treatment with both vectors actually. So it seems to work. And again, also other markers which were absent, like GPR179 or severely reduced, could be restored in the tiny dots here. And another nice example where it's not so much background is also GNB5 proteins, which are regulatory proteins, which are absent in GRM6. And here, after treatment, you have nice restoration of the protein. Unfortunately, it might be that this was not efficient enough, because we didn't obtain an ERG rescue after 2 months, nor after 4 months post-injection, neither under scotopic nor photopic conditions. But we continued our approach by treating LRID3, changing a bit the strategy, because the expression of LRID3 is not so sure. Is it only on the dendritic tips of the bipolar cells, or due the effect in the cone on bipolar cells? It might be that LRID3 is also localized in there are other studies showing this actually, that it is also localized in cone photoreceptors. So we changed our strategy. We targeted either the photoreceptors with GRK1, so promoter targeting rods and cones, and again targeting the bipolar cells with this GM6 promoter. Or we mixed the two vectors targeting photoreceptors and bipolar cells together. And this revealed that LRID3 was present after treatment in the bipolar cells or in the outer plexiform layer. So we don't know at which side actually, on the cone side or rod side or on the bipolar side. So let's say in the outer plexiform layer, when we targeted bipolar cells, when we targeted outer plexiform layer, as well as when we targeted photoreceptor cells. Even the best result was obtained when we targeted the photoreceptor cells. Similarly, we restored also LRID3, TRPM1, sorry. There were more dots than in the knockout mouse after treatment. And then the question was LRID3 restored at both at the rod on bipolar cells as well as on the cone on bipolar cells? These are the bars here. These are the cone bipolar cells, and the other ones are the rod bipolar cells. And when we co-stained with mGluR6, it looks already that it's mainly the rod bipolar cells which are restored. And this was further confirmed when we use cone arrestin. So here below in red yellow, this would be the staining of the cone on bipolar cells. But even after treatment, we don't have these red yellowish staining below of arrestin. However, under scotopic conditions, we nicely observe B waves by either treating here in blue bipolar cells, both bipolar cells and also photoreceptors, and the best results were indeed obtained when we treated or targeted photoreceptor cells. Again, no rescue under photopic conditions. However, the night blindness, which is the effect, the main effect in patients with CSNB, this phenotype was restored and stable even after 4 months after treatment. This was the time where was COVID and Juliette couldn't go to the lab, so this was good that she had to leave the mice for 4 months there alone, and they still had their B wave after this. And then we performed also the behavioral tests by using optomotor response measurements. And again, the scotopic responses were restored, not the photopic ones. And also we performed together with Serge Picot's lab on ganglion cell responses measurements by multi-electrode array studies. So we show here, this is from the wild type mouse on responses. So it's on the ganglion cell level. We want to see if the signal goes through the retina. That's not only the bipolar cells which can function now, but also the ganglion cells. So in the LRID3 mouse, there is mainly no B wave and no on response here. And after the treatment targeting bipolar cell, it looks like that there is some on responses, but they are really delayed. Better results were obtained with when the outer plexiform layer, so bipolar cells and photoreceptors were treated, and the most striking results were obtained when photoreceptors alone were targeted. So this brings me to my take-home message. So for the GM6 project, we showed that the protein localization in CSNB was possible in adult mice after treatment. However, the absence of functional vision after treatment was still recognized. So this may be due to the low transduction during these experiments. For LRID3, we had possible protein localization as well as in functional restoration after the treatment in adult mice. So at least the night vision was partially restored. However, the photopic vision after treatment was still not recovered. So, and it seems so that it's quite challenging to target LRIT close to cones. So in general, we concluded that to target bipolar cells, it's very tricky. And new promoters, I think, or AAV vectors need to be generated. Denise told me she wants to work on this. And, but however, also the administration may be a point to be changed. So perhaps we should also try subretinal instead of intravitreal injections. And then one point is that we thought that it's always okay to treat adult mice, but however, there may be a it's a synaptic defect, so there may be a missing synapses, and therefore it may be more important to treat at younger ages than in adult mice. And as I mentioned before, when Jan presented, there are also species differences and they may play a role. So perhaps changing the model may be also of that advantage. Yeah, there were a lot of patients I like to thank here and implicated, especially in the gene identification studies. Without this, the studies of therapy, of course, could never have been done. A lot of people involved in the patient collection, sending me the DNAs. A lot of people involved in the therapy study, especially Juliette, and platforms at the Institut de la Vision, the imaging platforms, and of course the animal platforms, and our team from A to Z, so Isabelle Oudot and myself as team leaders. And I think this is one of the last pictures we made before the COVID crisis. So thank you for your attention.
Thank you very much for this great presentation, Christina. This was very nice. You had beautiful results, I think. And because I'm always interested into the AV, it's nice to see a good serotype that's working quite well. I see that Alex has raised his hand. Maybe you can stop sharing so we can.
Oh, sorry.
Yes, it's okay.
Thank you very much for the presentation, Christina. I had a question. Well, you already mentioned that maybe the treatment needs to start earlier, but it was more whether eventually this could be corrected, even if it's targeted from the beginning, or it can be something more developmental that cannot be arranged, that the connection between photoreceptors and bipolar cells is made properly. Yeah, so I don't know, what's your opinion?
Yeah, I agree. There's one group in the States, and they made actually a transgene approach, and they had nice B waves everywhere. So I agree completely with you. I think there's also a developmental problem. And if you see where these genes are expressed, I think we need to check their expression earlier in embryonic stages even. And so it seems to be more tricky than we initially thought. We thought, oh, it's a stable disease, so easy to target adult mice, but no.
Thanks. And the other question I had is because you target photoreceptors and bipolar separately. So for the second part of your talk, the LRIT3, did you try to target both at the same time?
Yeah, exactly. This was the best results were with photoreceptors. The second best results were targeting photoreceptors and bipolar cells. Okay. And the worst results were with the bipolar cells. It was tricky, but for the photoreceptors, we got the best results, but this means perhaps as well that LRIT3 is expressed as well on the cone sites, and therefore it is the correct target. At the end, we think that anyway, where you treat, perhaps it's just important that it localizes correctly. So perhaps it doesn't matter if we treat photoreceptors and then it goes to the outer plexiform layer. Perhaps this will work as well for embryonics. I don't know.
Yeah, thanks.
And
Thank you.
just to continue on this, because I may have missed this, but with the last approach, you haven't tried the CAG promoter. It was always a specific promoter, either for the photoreceptor or the bipolar cells.
Yeah, it was with this we had good results, because I think I need to check again, but when we saw this distribution of the CAG and with TRM6, I think we thought, and we understood that LRH3 may be also expressed in cones. We thought, it's even easier to treat photoreceptors, than bipolar cells. So we want to be specific.
Yes. Okay.
Yeah, bipolar cells is really a mess to treat.
Okay. Thank you very much.